How stable are these changes in methylation, these methylation patterns that are so-called the aging clock?
Yes.
How stable are they over a person's lifetime?
I mean, they are remarkably stable. So when we compare to any other genomic measurement, I mean, they would be far more stable than anything I'm aware of. They're more stable, far more stable than gene expression, proteomics, metabolomics measurements. All the omics are less stable, And that's really the biological reason why these epigenetic clocks are the most accurate measures of aging. It's just that methylation is so stable, And it's stable not just in vivo, but also when people collect DNA, so we've collected DNA and then we didn't store the blood tubes properly, so they melted, and we extracted DNA and the measurements were perfect, So even on a technical level, they're very stable.
That's interesting, because I've done experiments intentionally, though, almost the same, where we were collecting blood samples from participants in a trial, and I was measuring DNA damage as biomarked by gamma H2AX. And I wanted to know how long I could have blood at room temperature before I started to get artifactual DNA damage happening.
Yes.
And so I did time course and, found that after 2 hours of blood being at room temperature, there's just tons of DNA damage that started to increase.
Yeah,
that
that's
was
scary. That's
right.
Scary.
So
that wouldn't be the case with DNA methylation. I know, because I hired a phlebotomist here in LA to visit families to collect blood, and this person didn't have an air conditioner in his car, and it was the hottest day in LA ever. So all the blood tube melted. That was my experiment, and then I just couldn't send this phlebotomist out to go back to the families to collect blood. I felt sorry for the family, so that's why we did this experiment.
Yeah.
And all I can tell you, we got beautiful data, and so I see that over and over. People sometimes ask me, what if you have a forensic sample? So let's say a blood stain, that was, let's say, in a room for a couple of weeks, or even let's say a bone sample collected, a couple of months later. All of these data, in my opinion, would still lead to very good methylation measurements for the purpose of measuring aging.
Yeah, because it's so stable.
It's just so stable.